All right, so the first video in this folder, the non-Mendelian inheritance patterns, codominance and incomplete dominance. One thing I didn't say, and I hit stop, was that these are often confused. So make sure you understand the definitions, right? That you get a third intermediate phenotype with the heterozygous individuals that makes them completely different than the two homozygous conditions. Okay, so again, think paint, mixing red and white paint, you get pink. Right? In complete dominance, you have some of each of the homozygous phenotypes being expressed, right? So in the case of the black and white chickens, you get some feathers that are black and some feathers that are white. The feathers are not gray. If they were gray, that would be a good example of codominance, okay? So that being said, those are the first two of the non-Mendelian patterns. This next one, okay, is called multiple alleles. So this one gets a little bit more complicated, all right? So the only example that we're going to talk about is human blood typing, and specifically the ABO blood groups, because there's another component of our blood types that's mentioned down here in red, okay? So this is called multiple alleles, right? When we talk about the other, let me start with actually the other component of our blood type. When you talk about blood types, you have either an A-type blood, a B-type blood, AB, or O. And then you also have a positive and a negative, okay? Notice this says positive and negative is a separate trait that does follow Mendel's rules. It's called the RH factor. So individuals that have two dominant alleles will have a positive component of their blood type. Individuals that are heterozygous will also have a positive component. And then individuals that are homozygous recessive are those with a negative blood type. So for example, I have type O positive blood. Now, I don't know if I'm heterozygous or if I'm homozygous dominant. I don't have a clue, okay? But that being said, I know that I have a positive blood type as far as the RH factor goes, okay? So that's a totally different trait than what we're about to talk about here. So for multiple alleles, first off, in the human population, there are 3 alleles for blood type. And they're written here, okay? This is how you write them, right? An I with a capital A, capital I with a capital A, capital I with a capital B, or a little I, right? And these I's stand for immunoglobulin, and that deals with antigens and antibodies, some things that we'll kind of talk a little bit about here in just a minute. Okay, so this is determined by the presence or absence of a certain molecule on the surface of the blood cells, specifically the red blood cells, all right? And that molecule is called an antigen, okay? And so I'm going to write that over here. These molecules are called antigens, okay? And so our cells are either marked with these or they're not, just depending on what your blood type is. Right, and so this is, just as far as some examples, this is really important for blood transfusions, paternity testing, some other things we'll talk about, but those are just a couple of examples. If you don't know what paternity testing is, that's where if there's a woman who does not know who the father of her child is because she's been promiscuous, then she might choose to have a paternity test. I won't talk about that tons for right now. Not important for what we're doing. So anyway, that being said, the IA and the IB are codominant. So they're going to follow the same pattern like the black and white chicken essentially. But they are both completely dominant over the little I. So they're going to follow Mendel's rules if present with the little I. Okay. So then on the next slide, and then we'll work the problem. Okay. So on the next slide, you've got your phenotypes. Okay. You can be A, B, AB, or O, right? People that are A can either be homozygous for A or they can be heterozygous for A, and that A covers up the little I allele, follows Mendel's rules, okay? People with B are the same way. They can either be homozygous for B or they can be heterozygous for B. People with AB have an A allele and a B allele, one from each parent, so those are, again, codominant. And then people with type O, like I'm type O, have 2 little I alleles, okay? Now, what does this part mean? So this means that people with type A have A molecules on the surface of their blood cells that are called antigens. People with B have only B molecules, and people who are type AB have both, okay? And then people with type O, we have no markers or molecules on the surface of our blood cells, right? So from a blood transfusion standpoint, people with A can give to somebody with A or to somebody with AB, okay? people with type B can give to somebody with B or somebody with AB. People who are type AB can only give to other people with type AB, right? And then people that are type O, because we have no markers on our blood cells, we can give to anybody, okay? So there's nothing that's going to be seen as a foreign molecule to anybody else's type blood if I were to give them type O. Take the RH factor out of it, the positive and the negative, okay? So I can give blood to anybody because my blood is not marked with these A or B alleles, right? Or chemicals, the antigens, okay? Notice, though, I can only get a transfusion from someone with type O specifically, okay? And that's because the A antigens and the B antigens are going to be seen as foreign, right? So if I were to be given the wrong type of transfusion, it can cause what's called agglutination, which is when your blood clots, and it can actually kill somebody. So this is really, really important, okay? For somebody with type A, they can get blood from somebody with O or somebody with A. B, they can get blood from somebody with B or somebody with O. These can't swap because B antigens are foreign to people with A and A antigens are foreign to people with B, right? And then people with AB, they can take blood from anybody because A antigens are not foreign, nor are B antigens, nor are the lack of antigens on type O. Okay, so I am considered the universal donor, and somebody that's AB is considered what's called the universal recipient. Okay, so that being said, right, and this just gives you some kind of breakdown, and I don't know if this has shifted any from these percentages, but this is pretty close, okay, as far as the percentage of the U.S. population that has these different blood types, right? And so then, for example, too, since I'm O positive, I can actually have blood from somebody that's O negative, but somebody that's O negative cannot have my blood because that positive would be seen as foreign, and it would cause the bloods to clot as well, okay, just like when you're looking at the antigens up here. Okay, so let's look at our example here. So we've got a man with type AB blood, and he marries a woman with type O blood. What are the possible genotypes and phenotypes for the children? All right, so then we're going to follow our typical rules, but because it talks about man versus woman here, let's make sure we put our alleles in the right place. So the man has AB blood, and the woman has type O blood, okay? So then this box is going to be IA little I, this is going to be IB little I, IA little I, and IB little I, okay? So our genotypic ratio is going to be 2 that are heterozygous for type A to 2 that are heterozygous for type B, okay? So none of their children are going to have the same blood type as either parent, okay? And so the mother could donate blood to one of her children just based on the ABO blood groups, but the father could not, because again, this A antigen is going to be foreign to these kids, okay, that fall in this half, the B antigen foreign to any kids that fall in this half. All right, and so then the phenotypic ratio, that should be a P, okay, 2 that are going to be type A boxes and 2 type B, okay, and of course both of those will, you can't see that, sorry, both of those will reduce to 1 to 1, okay. All right, so then your last type of non-Mendelian inheritance pattern that we can actually put into a Punnett square is called a sex-linked trait, okay. This could also be called, but it is not, a gender-linked trait. So this is not linked to like the act of sexual intercourse. This is linked to gender, male versus female, right? So when we look at what's called a karyotype, and with me recording this right now, I am not sure if we will have talked about this in class yet or not. We'll just kind of have to see. But when we look at our 23 pairs of chromosomes in humans, the first 22 pairs are called autosomes. The 23rd pair, that's the sex chromosomes, right? So that's the pair that it determines the individual's gender, right? somebody that has 2 X chromosomes is female, and somebody that has an X and a Y chromosome is male. And this is a little Punnett square kind of turned on its side, okay? So that remember, for every round of meiosis, we're going to get 4 cells, 4 true sperm. 2 of those will have a Y chromosome, 2 an X. And then for eggs, we're going to get 1 egg and 3 polar bodies, but we still write this the same way, okay? So that any any children in this part of the Punnett square are going to be females, and any in this part of the Punnett square are going to be males, right? So it says right here, this particular bullet, males determine the gender of a child. They're going to donate the Y chromosome to a son and the X chromosome to a daughter. So if you are female, your dad gave you an X chromosome, and so did your mom. If you are male, your dad gave you a Y chromosome, right? Your mom still gave you an X chromosome. Okay, so females, we only donate X chromosomes to our children. So anything that we say is a sex-linked trait is a trait that's controlled by the genes that's specifically on this pair of chromosomes, right? Now, the Y chromosome is really tiny. So the traits that are associated with the Y chromosome, it's just basically things that truly make males males. All right, when we talk about sex-linked traits, the ones that we're talking about are those that are attached to the X chromosome. Okay, so this is just one example of how to write an allele. All right, so it says, therefore, females are going to have 2 copies of the alleles. Okay, so basically for a female, we've got one example here. If that's a recessive allele, a female could be homozygous dominant, a female can be heterozygous, and a female can be homozygous recessive. Okay, so that's females. And then males, and I'm just using these same alleles. It doesn't really matter what we use for what I'm doing right now. Okay, a male can have a dominant allele, and a male can have a recessive allele. Okay, males only get 1 allele for this type of trait, and so they get the allele whatever their mom donates. Okay, so because of that, sex-linked traits that are recessive are much more common in males than in females. Because look here, if a female gets 1 dominant allele and 1 recessive allele, then that dominant allele is going to follow Mendel's rules and mask that recessive allele. All right, and so then as far as examples go, okay, and then we'll work a problem. All right, so these are a couple of examples. So color blindness, and just based on me doing this as a recorded lecture, I don't really know if I have any of you guys that are colorblind. I've had a few students over the years that have been. So here, for example, if you can see that really easily and tell that that's a 25, and if you can pick out these other numbers, you're probably in pretty good shape. People who are colorblind, typically the most common form is red-green color blindness, and people that have it can't necessarily tell that this is number 8, okay, for example. All right, male pattern baldness, this is the balding pattern, okay, that is somebody gets older and is losing hair, just because that tends to happen as we age, okay, that if they have that little ring of hair around the back of the head, like my stepdad has this, and the top of his head is bald, then that's considered male pattern baldness, right? Another example, so this is the Russian royal family. And hemophilia is an example of a genetic disorder that this family, the son, Alexei, he had this family, hopefully you'll learn about with the Russian Revolution in history. If y'all do, we'll kind of talk more about it. But anyway, that's an example as far as why that picture is there. So he's the one that was considered a hemophiliac. And hemophilia is a genetic disorder where the blood does not clot normally. Now people take clotting factors for that. There's a lot more I could say, but I'm not going to do it as far as this lecture goes because it's already getting long. And then here's another example, fruit flies. The color of the eyes in fruit flies is either white or red, and that's a sex-linked trait. Okay, so a non-human example. All right, so let's look at our example of color blindness. So it says color blindness is an X-linked recessive trait. Determine the genotypic and phenotypic ratios of a cross between a woman who is a carrier. Okay, so that's going to be carrier. This is going to be somebody that's heterozygous. Okay. And when we talk about genetic disorders, the term carrier is much more important, whether it's a normal Mendelian trait or something that's sex-linked, for example. right? So we've got a woman that's a carrier for color blindness and a man with normal color vision. So let's use a capital N for normal color vision, and let's use a lowercase n for being colorblind. Okay, we could really use anything. Okay, doesn't really matter, right? And so this type of inheritance pattern is going to trip more people up than any other type, right? If you just use n's, your answers are automatically wrong because this is sex-linked, so you have to include the sex chromosomes. All right, so we've got a woman who's a carrier, so we're going to write her genotype like this. Okay, so she's heterozygous. And then the man, what does it say again? The man has normal color vision. So we're going to write his genotype like this. So that's our cross, right? So then when we put that into a Punnett square, and I'm probably going to have really crazy spacing for some of this. So we're going to have our male, because we know they go along the top when we know information about male versus female. There's the female's genotype, okay, split up into gametes. Okay, there's our first box, our second box, our third box, and our last box. Okay, so from a genotypic ratio standpoint, and I'm going to have to write these just because I'm running out of room. Okay, I'm going to have to write them where it's going to say we're going to have one that's the female that's homozygous dominant, okay, to one, and I'm going to write the females together, okay, and write them, sorry, back to back. I shouldn't really say together. Okay, to one that's homozygous, no, sorry, to one female that's heterozygous. Okay, so pretend this is all on one line. To one male with a dominant allele, to one male with a recessive allele. So from a phenotypic ratio standpoint, we're going to get 2, this and this are both going to be normal color vision, but in females. So 2 normal, and I'm just going to write 2 normal females to 1 normal male, to 1 colorblind male. Okay. And guys, if you truly just used N's, okay, and made that male have 2 alleles and tried to do it like a normal Mendelian inheritance pattern, then it's automatically wrong, okay? There's nothing I can do for you. You missed the entire problem, right? All the time because you didn't even begin to tell me that it's a sex-linked trait. One more thing about this. So for sex-linked traits, only females can be a carrier, okay? Males are either going to exhibit the dominant allele or exhibit the recessive allele. If we're talking about a trait that follows Mendel's rules and it's just a simple, let's just use B's, I don't know, it doesn't really matter, okay? We're talking about some trait that follows his rules. These are your carriers, okay? It's your heterozygote, I can't spell, heterozygous carrier, okay? So like cystic fibrosis, for example, is another genetic disorder that is going to follow Mendel's rules, and so a carrier of that would be written like this, okay? Hang on. Okay, so then our last example of non-Mendelian inheritance patterns, these are called polygenic traits. So remember, poly means many, so these are traits that are controlled by more than one gene, right? All the other traits that we've talked about so far, whether they're non-Mendelian traits or they are traits that follow his rules, they all are controlled by a single gene that's always at a single locus. Well, a polygenic trait is going to be controlled by multiple genes and multiple loci, and so these are going to exist on what's called a continuum, okay? For humans, skin color and height are 2 great examples of this. So not that we would do this in class, okay, but we could do with this, this is probably like a professor from a genetics class like in the 70s. If you look at this picture real closely, like lots of bell bottoms and whatnot. So probably lined their class up where most people are of average height, okay? Very few people are really, really short, right? Say like 4'8", okay? Very few people are really, really tall, 7 feet tall, okay, in the grand scheme of things. Okay, we could also do this with skin color. we have a continuum in our class going from people that are really light complected like me to people that are much more darkly complected. And probably if we kind of looked at the overall proportion of those, most people kind of have intermediate skin pigmentation. All right, this is another example that's not a human example. This is bell peppers, okay, and that's controlled by a polygenic trait, the different colors that we can see there. All eye colors are considered polygenic except for blue. Blue's completely recessive to all other colors of eyes. That's why you see some kind of differences with different shades of brown. Hair's the same way, okay, hair color is. There's some specific things about being a redhead. There's a specific gene mutation that causes red hair, but otherwise hair color is polygenic as well, okay? So we can't plug these into a Punnett square, okay? Again, they're going to exist on a continuum, right? And that we have vastly different kind of extremes or ends to that continuum, okay? Tons of variation, right? Which is, you know, the variation that we see as we walk our halls every day. Okay, and then the last thing, and I'm going to cover this on this particular PowerPoint, this particular lecture, just because it's printed in the same PowerPoint, is what's called a pedigree, okay? And if we have not covered this already in another way, just kind of bear with me on that. Just hang on just a second, though. Actually, you know what? Never mind. I'm going to put this on a separate PowerPoint. If you have any questions going back to polygenic traits, multiple alleles, sex-linked traits, codominance or incomplete dominance, let me know.